talk. Okay. So great to be here virtually with you and to be able to travel the world without flying for 20 hours, though I'm sure it would be more fun to be in person. I'm going to talk about genomic epidemiology in SARS-CoV-2 and some of the methodological challenges that we're thinking about. So I'll just jump right in. First off, what is genomic epidemiology? I'm a mathematician in some sense, although I do very applied stuff, but we like to start with definitions. And the way I'm using this phrase is to build methods and use them to use pathogen sequences to understand transmission of a pathogen, here a virus in this case, obviously. So transmission can mean different things to different people. It could mean global geographic movements of the virus. It could mean past population dynamics, how much virus there was. People use sequence data for this. And it can also mean like really drilling down into person-to-person transmission, who infected or did not infect whom in an outbreak. Sequences in principle offer a high resolution view of how infections spread, right? Because in order to have descendants, to have offspring, infections have to move through populations. And as they do that, they pick up mutations. And so we can really get a sense of their shared ancestry with sequence data. And that gives us a sense of how they spread and how they're transmitted. But that resolution isn't perfect. And that presents some challenges. So basically, one of the key ideas is this. Suppose we have our pathogen and we sequence its genome. So we read the RNA of the virus or the DNA of the bacteria, however it is. We can use this to estimate how closely related people's infections are. So if they have these two sequences, they maybe differ at a few points and they're very similar. But that tells us something that, these two individuals who gave rise to virus sequence 1 and virus sequence 2 had closely related viruses. That's great. That doesn't jump off the page and say, okay, Alice infected Eve and Eve infected Bob and Bob infected Fred, right? And it doesn't tell us, how rapidly transmission is happening or these other features that we might want to understand. So I think one of the core tasks of genomic epidemiology is you get these sequences and you try to understand something about epidemiology and transmission from them by making inferences. Okay, and this is a schematic view of, I guess, how I'm seeing it really from the point of view of this talk. Here's a phylogenetic tree. The little dots are virus sequences and the edges are represent inferred ancestral relationships among the viruses. We might find some closely related little clusters where we think we can really drill down and understand something about transmission. So we might want to do that. And then having done that, we maybe say, well, we don't know whether Alice infected Bob and Bob infected Eve, or whether Alice infected Eve and Eve infected Bob, and so on. But we might want to understand some patterns that we can generalize from those possible infection paths and relate that to transmission dynamics and forecasts. And I think this is an area where genomics have been underutilized. We have a lot of great genomics papers that exist that are very retrospective in nature, building and coloring in these objects and understanding the selection and evolution, and fewer that say, okay, what does that mean in terms of what's happening next? We don't get to what's happening next in this talk, but that's kind of sets a context for the transmission dynamics that I want to talk about. Okay, so one of the goals, or the main goal of this first part of the talk, is to use sequence data to help us estimate the serial interval or the distribution of the serial interval in a way that's kind of high resolution. And by that, I mean specific to a particular group of sequences, an outbreak, or a lineage, rather than say, okay, well, COVID, it's 5 days-ish, 3 to 6 days. We say, well, in this context here, could we figure out what the serial interval is with the help of virus sequences? Okay, so what is the serial interval? Here it is here. We have, in this case, 2 people, and A infected B, and A infected B at this time. After an incubation period, B got symptoms, and A got symptoms too. And the difference in time between their symptom onsets is the serial interval. It can be negative. It could have been the case that A actually didn't get symptoms until after B. We're going to ignore that here. It could even be ill-defined because might be the case that someone doesn't get symptoms at all, and we're not going to worry about that either. Or we could say, well, instead of symptoms, we're going to talk about collection dates of the sample, and then everyone in our sample, by definition, had a collection date. That's how they got into the data. Okay, so why do we care about serial intervals? Serial intervals are really important in modeling infectious disease transmission. They help to estimate how much transmission happens before symptoms. And I didn't say that here, but this green part is how long before A got symptoms did A infect B? And that's really important. And it's one of the defining features of this pandemic, that transmission happens before symptoms too much, so that we can't control the pandemic by isolating people who have symptoms. That's one of the huge differences between this virus and SARS-1. So if we can estimate the serial interval and we can estimate the incubation period, we can figure out how much transmission happens before symptoms. Did I hear a question? Nope. Okay. Someone shout at me if you can't hear, or if there's something unclear, just let me know. Or if I freeze, because I can't really tell if I'm frozen to you, because I can't see anyone moving to tell if you're frozen to me. Okay. So serial intervals are important. They depend on the pathogen and on the population. They are used in estimating the basic reproduction number. And therefore, they are used in estimating the portion of the population that needs to be immune before cases start to turn around. They are used in all those estimates of RT that you see on Twitter, and they are fundamental, underlying parameters for nearly all modeling efforts where we think about what's going to happen if we wear masks or whatever. But one of the issues here is that there are very few sets of data available to estimate serial intervals, and so there are relatively few estimates. Conventional estimates of serial intervals are done by finding some pairs where we think A infected B. That's hard because you need contact data, you need really boots on the ground epidemiological investigations. But find those pairs, working out how long it took between A's symptoms and B's symptoms. Doing the statistics to account for the fact that if A infected B and B took a really long time to get symptoms, you might not have seen B yet, and so you don't know who A exposed, and so there's a right truncation problem that you can manage with statistical methods. And then proceeding to figure out all these times and estimate how long it takes between A and B's serial intervals. We wrote a paper like this. There are many others out there, and that was one of our first pandemic papers, was to use publicly available data from Singapore and from Tianjin, China, to do this kind of analysis. And here are some others, and you can see them here. These are the estimates and confidence intervals. And all of these studies use contact data, so some data describing who infected whom, and when their symptom onsets occurred. Okay. So we're going to use I'm going to describe a new method that uses genetic sequences in the way I described to, as kind of a proxy for detailed contact tracing data, with the aim of, I skipped a slide by accident, with the aim of improving our ability to estimate serial intervals and making better use of genomic data. And one motivation for that is that sequencing the virus is now done routinely. So there's all these labs who are routinely sequencing some reasonably good portion of the infections they detect. And we can say, well, okay, maybe we don't know exactly who contacted whom and who infected whom, but we can use the genomes and the timing and the place to identify some plausible transmission pairs. And then we can use those plausible pairs to estimate serial intervals. So in this way, we connect the sequence data from the virus to methods to estimate serial intervals. Okay, we're using data from you good people in Australia in a collaboration with Melbourne through Courtney Lane, Ben Howden, and Watt, and others. Anders Gonçalves da Silva, I don't know if you know him, he lives in Canada, but helped us made the connection between our group and you folks. We have data from your wave 1 and wave 2 in Victoria, And this shows, I'll talk about the clusters more later, especially if we have time, but this shows the cumulative cases divided up by genomic cluster in wave 1 and wave 2, where there were more cases. And of course, you probably know more about your local epidemiology than I do. This is a phylogenetic tree from the wave 1 data, and it shows, so again, these colored dots are virus sequences, and the edges, the black lines, are inferred ancestral relationships among those. And we've just colored in the different clusters. So you can see they kind of group nicely genomically. And these form the units of analysis on which we want to estimate our serial intervals. Okay. So here's the approach in brief. We're going to start with these clusters that I just showed you. Here's another schematic of the same kind of thing. These ones happen to be from BC. And then we're going to identify these plausible pairs using our genetic distances and differences in collection times or symptom onset times. And then from those pairs, we're going to sample plausible transmission trees. Those are all stories of who might have infected whom. For each of those trees, we're going to estimate the serial interval distribution in a way that we've made up that I'll describe. And then we're going to combine those estimates. And so our confidence intervals have to incorporate the fact that we don't know who infected whom here. And because that's, this isn't the boots on the ground, really careful epidemiological investigation. This is this easier kind of data to get. So we have to take that uncertainty into account. Okay, so here's the schematic that summarizes some of the work I'm not going to talk about. I'm not going to talk about the sequencing methodology and the bioinformatics that go behind this. I am going to try to talk about new approaches to clustering, but not in the first part of the talk. This part of the talk is focused on these two boxes. Get the transmission cloud of all the possible who infected whom stories, and then use them in a statistical model to estimate serial intervals. Okay, so now I'm going to tell you about the statistical model that we use to get our serial intervals out of all these times and pairs that I'm talking about. So we're going to cover 3 ways that we could have. So we're going to get our pairs from our genomes and our times. So we're going to say, if you guys are close, your viruses are really closely related and your times are compatible, you guys are, congratulations, you guys are a plausible pair. And that's these plausible pairs of these 2 red cartoons in the top corner. Now, it might be that the plausible pair is really, A infected B, and that's what we want. We would like, ideally, we would know all these who infected whom, and then we just look at how long it took, right? But we don't actually know that. So, but in this case, imagine that we did. And then we're going to say, well, our serial interval is gamma distributed. And so that gives me a distribution for, how the time will look. And that gives me a likelihood or a probability. And that has 2 parameters, a mean and a standard deviation. And it kind of looks like this. But of course, it might not be that A infected B. We just have these plausible pairs. It could have been indirect, right? And I'll talk about this coprimary in a minute. I don't think I missed a slide, did I? No, I didn't. Okay, so it could be that we have this indirect route where we have A and B, the 2 red cartoons, but in between them, actually, there were some gray cartoons that we missed. We didn't sample those people. So A infected unknown, 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 and some series of unknowns, some geometric random variable number of these unknowns. And then instead of okay, so that's possible. And then what happens? What does that do to the time? Well, the time between red symptoms on this side and red symptoms on the other side, the 2 symptom onset times, becomes not just 1 serial interval, but actually you have to go through all these intermediates. So you get a sum of these random variables with our distribution. And it turns out the mathematicians knew we were coming, and they've worked out like what all these distributions look like. And this one is a compound geometric gamma. So it's a sum of a geometric number of gamma distributed random variables. And that turns our blue single peaked distribution into this red distribution where, oh, look, like there's your main thing. And you might have had an intermediate, or you might have had a few intermediates, and it goes off and has a longer tail. Okay, so now we've had A infects B. We've had A infects some number of unknown people before getting to B. And then finally, we might have had this route, this so-called coprimary route, where actually A and B, rather than infecting one another directly or indirectly, they were both infected by the same unknown person. And that can make their apparent interval look really short, right? because they might have symptoms six hours apart or a day apart. And it's not that one infects the other and it's a one-day difference. It's that actually they were both infected 4 days ago, and one showed symptoms today and one showed symptoms tomorrow. And in that case, if we take the later symptoms and we subtract the earlier symptoms, we always have a positive number. And that's that distribution is also well defined, and people have worked out what it is. And it's a folded gamma difference distribution, so a difference between two gamma random variables. Okay, so that gives us actually a whole statistical model for the set of time differences that we're going to have when we choose these plausible pairs. So we have this whole set of TIJs, the time of symptom onset between I and J, of these pairs of red cartoon folks in our data. And we have this model that's like with some probability, it's this compound geometric gamma distribution, and with some other probability, it's this just gamma difference distribution. And there we have these 4 parameters that define a statistical model that relates our serial interval that we want to the times that we observe. Okay. So here it is on one slide: direct, indirect, or this co-primary, and that gives us the statistical model. And we estimate the parameters of that model using a kind of minimalist Bayesian approach, where we have beta priors for these 2 parameters, W and pi, and then we take a maximum posterior probability to estimate those parameters. Okay, so far so good? Somebody want to nod? Somebody nodded. Okay, thank you. That's great. Okay, so that's what you do once you have a bunch of pairs of red cartoon individuals. We get those plausible pairs using our virus sequence data and our times. So a plausible pair, 2 people are close enough in their symptom onset time that it's feasible one infected the other through one of these routes, or their collection date, and their viruses have to be close enough genetically. So if there were 50 polymorphisms between 2 individuals, we would say, nope, that's not direct enough. If there were 3 or 4 or 10, we're probably going to say no. If there are 0 or 1, we're going to say yes, and we're going to select with higher probability if the 2 individuals are genetically closer in the viruses. And we will also select nearby times with higher probability. We then make zillions and zillions, I think about 100 in practice, of these plausible who infected whom stories, and then we go ahead and use them all because they're all plausible. So we run this model and estimate these 4 parameters with all these different who infected whom stories. Here are some of the visualizations of specific who infected whom stories that we get. And on the left-hand side with the yellow-orange nodes, we just have case IDs and edges that are some of these plausible events. And then the gray numbers indicate the number of days between their symptom onsets. So you can see here's a 16, maybe likely that there's 1 or 2 intermediates here, or 4 or 5. And there's some 1s here where they got symptoms the next day. And maybe those are those co-primary, both infected by an unknown individual. And then if we want to, we can sample lots and lots of these things and make a different kind of graph where we take the nodes are cases and the edges are links, these plausible links, but they're weighted according to how probable they are, how many times they appear in this random sample. I should say, if I really thought that this was a great, way with SARS-CoV-2 to really define transmission networks, I wouldn't be focusing just on estimating serial intervals, right? I'd be focusing on, look, okay, let's tell stories about transmission. But I think these things are really quite uncertain because SARS-CoV-2 doesn't actually mutate fast enough that we can really define transmission events as individual events with high probability. But we're sort of pushing the boundaries here, and for a more mutating virus, or maybe if we want to interpret this as kind of indirect transmission under weaker sample, we could do it. But I wanted to put out that caveat right now. Okay, so what do we get? What happens? Well, here are some of the results for the mean and standard deviation of the serial intervals from the Wave 1 data. And you can see they vary a little bit from cluster to cluster, but I wouldn't like hang my hat on like, oh, look, this one is small and it's significantly smaller than these other ones, because they are really quite widely varying. And in a way we wouldn't really expect dramatic differences, as this virus arrives in a fully susceptible population in early 2020. And we don't know really who infected whom. These are in line with international estimates of around 5 days. Maybe some of them are a little shorter. And so, that's great. That's in a sense, it's a kind of coarse validation of the method that we're getting these sensible answers out. It gets a little more interesting when we look at the wave 2 data. With the wave 2 data, I'll show you a better plot. If you just use time and genetic distance to divide the sequences up into units of analysis that are clusters, it actually gets kind of unwieldy. So in this wave, we used exposure sites. So if they shared a setting where both were known to have potentially been exposed or have had contact, that's different than using like really deep, we think that this person infected that person because they were both at the same restaurant at the same time and they blah, blah, blah. So it's sort of a shared context rather than really contact data. We use that to define the sets of plausible pairs. And here we get something maybe a little bit more interesting, although it's, again, I'm not necessarily going to hang my hat on, for sure this is significant. We found that the estimates of the mean time between symptom onsets ranged from 2 to 9.5 days. So quite a large difference in the mean from down here with a 2 in a cluster with a tag we don't know, to all the way up to a healthcare setting where the mean was really long and the confidence interval was really large. We did seem to see that meat processing, the orange ones tended to land more in the bottom part of this with shorter serial intervals, and more healthcare facilities and aged care settings tended to be on the high end. So notice that lots of these ones are red and very few of these ones are red. But again, there's a lot of variation here, and we haven't done an analysis. I suspect it would come up with a no, but if you asked, like, could you claim from this that the meat packing are longer significantly than the healthcare facilities, I would guess no. But we do find quite a bit of variation between the clusters. Okay, so I motivated this part of the talk by saying, these are so important because they change these reproduction numbers and they underlie all these modeling results, and they're profoundly important. They have all this impact. Okay, so you would be, very reasonable to say, okay, you've got these clusters, so show me that impact. Okay, so I'm going to show you a little bit of that impact on the effective reproduction number, which is the average number of new cases that a case causes over their infectious period. If that number is bigger than 1, it's growing. If they're less than 1, they're declining. If everybody's susceptible, it's the basic reproduction number. Okay, and estimates of that depend on the serial interval, which I've said at the start. So how do our estimates of the serial interval change anything compared to what you would get if you just use your international baseline standard serial intervals? And the answer is they don't change it very much. Here are a bunch of little clusters. And we've used the EpiStim package in R to compare the serial, the RT that you would get if you used the baseline gray versus the ones that we get with our new serial intervals computed in this cluster specific new way. And mostly it doesn't make any difference. But with several of the clusters, it actually does make a huge difference at the start of the cluster. I should say these things all die out because of the way we defined clusters. They have to end, and we had a finite time for our data set. So the fact that they all tail off doesn't mean COVID is over by August 15th of 2020 in Australia. I think we all know that. But at the start, you can see there's a couple of clusters where our estimates and conventional estimates really give you a different sense of the number of new infections per infection. And that's important because it impacts the speed with which you have to intervene if things are faster, and it impacts if you're looking at kind of immunity and when it might turn around, it would impact that estimate too. Okay, So you might ask, hey, you've invented two new things. You've invented a new statistical model with all these red cartoons and all these roots. And you've also invented a new way to get pairs, right? Instead of going out and doing contact tracing, you've done this genomics thing. And so how can we validate this? We want to understand, like, are you going to get the right answer beyond like, oh yeah, it's kind of similar to the 5 days that the international estimates had. So we've tried to look at this in two ways. One is if we did have contact data that was really closely, through which we defined many of the same pairs as we do with the genomes, do we get the same serial intervals? And also, if we just had, baseline data and we compared our new statistical model to conventional estimates, do we get the same answer? So here's one. So here is our estimates with our statistical model compared to other people's estimates with their statistical models. But we don't have a case where we have the same data, where we don't have genome data that exactly matches up with contact tracing data. So there's part of that validation we can't do. But here I'll say a couple of things. So published estimates use contact tracing data with this hard to get data, and they get these green, much tighter estimates of the serial intervals, right? Their confidence intervals are narrower. But those published estimates don't account for this possibility of co-primary transmission. They assume if you see A and B and it's a day apart, you have a day. So those contact intervals or those serial intervals are a bit shorter than ours, and they're much less variable because you have much better identifiability if you make that restrictive assumption. They also have better data. So they may have better estimates because they have better data that's much harder to come by. So in a way, this is kind of a validation. In a way, there are limitations to the extent to which this is a convincing validation. Okay, so that's the end of part 1 of the talk. I'm going to check the time. Okay, we're about halfway through. So I will talk about this next topic. Are there questions for this at this point? No. Or if there are, it's taking people a minute to unmute. Okay, I'm going to go. Does someone have one?
Caroline, I just, with the second wave in Victoria, there was certainly reported in the press that virtually, 99% of the cases were genomically linked back to one escape from hotel quarantine. Do you happen to know from looking at the wave 2 data whether that's correct?
I don't know whether that's correct, but it wouldn't surprise me that there's one founder event and then these clusters would be, diversified after that in these different settings.
Okay.
And I mean, I guess they're, even in a setting like here where there's more transmission, just by genetic drift, like it's sort of surprising how recently everything would have one common ancestor. So even if there were other introductions that all died out, it's still totally plausible that everything is traced back to the one event. It doesn't mean if that one event hadn't happened, you'd have nothing. Well, it might. I guess it depends how you structure your counterfactuals. Yeah, that's a great question. Okay, I'm going to move on. So we've talked about these clusters as if they are handed down to us. And in a way, this connects to your question, like where do these clusters come from? Why are these sensible units of analysis? This is an illustration from a genomic epi paper from India that illustrates contact-derived clusters. And this is kind of what I think public health agencies have in mind when they think about genomic epidemiology. You're going to get these sequences, and they're going to give you these really defined groups that are going to connect to contact patterns and social networks, and they're going to have this really like nice defined structure and provide these units of analysis where we can really meaningfully compare this cluster 4 with cluster 2. But why would that happen? How do we define clusters? Are they really meaningful units of analysis? Why would we expect clusters? Here's another one that's more phylogenetic, and it's a paper about HIV. This is also kind of how we might think of clusters in the field. You know, there's have this big tree that shows you the shared ancestry of all these HIV sequences, and you go, oh, look, here's a little clade, and this happened in my jurisdiction or in a particular setting, and I'm going to investigate this clade. And these other things are not in that cluster, and they're very genetically separate, and this is like a very clear story. But we thought, you know, if you have a human-to-human pathogen, and it doesn't go off and sit in a lake or something for some time and then come back into your jurisdiction, and it's really spreading in your jurisdiction where you're sampling, you might not expect a really clear population structure where you can really distinguish these meaningful units. So we were thinking about when that might happen, and developing ways to do clustering that addresses some of the challenges that public health faces, because there isn't always really this neat structure. Okay, so why would you not do, you know, what goes wrong with the picture I just showed? Why would you not make a phylogenetic tree from sequence data? There are lots of established ways to do this, and they're great, and they originated in the 60s and 70s, and they account for biological things, and they're really wonderful. We really think they're a great way to understand sequence data under conditions like we have for COVID. So why would you not make this nice object and then color it in and say a cluster is going to be a closely related clade or group on this nice tree? Just like I made this up, of course, in BioRender, and I colored it in. And on this tree, it looks nice. You have this blue cluster and this pink cluster. So what happens? Well, why not do that? That seems good. So one challenge is that the tree reconstruction is computationally expensive. In BC, we could sequence about 3,500 viruses a week. So as you go on and you get week after week after week, soon you have many, many tens of thousands of viruses, and making this one tree is expensive. But that's not such a huge problem. You could get over that problem. A bigger problem is that the genetic diversity may not be sufficient to create a really well-defined phylogenetic tree that you really believe in. And if you have an approach where you draw a tree and you color it in, but then you drew a different one that was kind of just as good, you might color it in a different way and get different clusters. And that's a problem because two cases that are clustered together this week, then next week I have 3,500 more sequences. And next week I then make a tree and I color it in. But now these two individuals who looked like they were in the same cluster this week are no longer in the same cluster next week. And that is not helpful if we want to understand clusters as something that means something epidemiologically, that we can send back out to health authorities in the regions where these outbreaks are taking place and saying, oh, look, cluster B76 grew from 3 or 10 cases to 27 cases. We don't want to say, well, it also lost 6 of the original 10 cases. This is not helpful. This just shows a random genealogy from 20 taxa under a coalescent model, for those of you who are mathematicians who've heard of the Kingman coalescent. And you can see that, you know, it looks like there's really distinct population structure here, with this clade being really separate from the rest of the, something biologically different might be going on here, but that's spurious. These trees can look like this in a neutral model. So if you combine these three things, there isn't really a robust way to do clustering. So we invented what we think is a somewhat more robust way that met these aims. So sequencing, people do phylogenetics and clustering, and then they want to inform outbreak analysis. In order to do this task, clustering should be feasible in real time. It shouldn't take 60 hours to make a tree. It should be stable week to week. And ideally, it should reflect some known epidemiological links. If you have really great clusters where you really did that hard boots on the ground work, you want them to appear in this clustering. Okay, so this is a schematic of what the BC's COVID looked like, and this is 2021. And it shows that we had lots of COVID compared to what you guys had. And we had lots of lineages. And we had, you know, up to, by the end of week 30, so August 2021, we had 30,000 sequences with quite a bit of diversity. So that inhibits, you know, every week making another big, high, you know, maximum likelihood computationally intensive tree. Okay, so how did we do this? We used a pairwise logistic model, just like logistic regression, where we modeled some probability or score, like the plausible pairs really, for two isolates to be linked. So where the links are going to ultimately define the clusters. And we just modeled that as, some offset and then a term for genetic distance, a term between the two, and a term for the time difference between the two. And if you like, you can make a term for the exposure site. So make it highly likely, more likely they share a link if they share an exposure site. You could make this region-based if you really thought people were not moving between regions. And of course, if you had restrictions, then people were less likely to move between regions. So you could allow for these other variables. But we used time and genetic distance because in BC, we did see a lot of movement around the regions. And we didn't have access to data about shared exposure the way we did for the Melbourne, the Wave 2 data that we had through our Australian collaborators. The nice thing about this is because it's logistic regression, you can train it with your known clusters if you like. You can say, well, I really like these ones, and I'm going to train my model and estimate coefficients. Or you can also use knowledge of the mutation rate and serial interval and so on to kind of guesstimate these coefficients using expert knowledge. How much does being closer improve the odds ratio of being a link, so that you can get that from literature. And then what we do is we say, okay, we're going to take edges between two cases, yes or no, according to a cutoff in P, and we're going to define the clusters as the connected components that we get. So if A is linked to B and B is linked to C, then A, B, C, and then nothing else, then A, B, C are a little cluster. Okay. So here is a phylogenetic tree, again, nodes and edges. With different clustering methods, our method with different thresholds on the value of the score with which we decide, okay, we're getting cut off. And then the standard kind of tree cluster, which is the draw phylogeny and color it in method. As you might expect, really variable cluster size with some huge clusters of potentially thousands of cases that are infeasible for outbreak analysis. A good sensitivity and for placing cases with known links together, so that's good. Cases without known links not being together is a bit harder because we know we don't know all the links, so there's some asymmetry in kind of judging these methods. Okay, so of course you can run a clustering method and you're going to get clusters. How good they are, how helpful they are, that's a little hard to tell. Our clusters, because of this logit model, they do not split apart when new data are added. And if you compare that to the tree-based clustering that people tend to use, they do have this problem, and 20 to 40 of the clusters will actually break up the following week as you add new data. And that's the thing that really the epidemiologists told us is completely not helpful. So our method doesn't do that at all. I guess one little blip happened over here and maybe one over here, but essentially that solves the disruptive problem of you make a new tree, all your distances change, all your coloring in changes, and you get this problem. So that's great. Okay, I will tell you, if what you want is a clustering that is really stable, you can achieve that in a trivial way, right? You can say, okay, everybody, you're all in the same cluster. Great, now I'm completely stable. This is going to work. But that's not useful. What we want is this idea of a kind of high resolution. So how evenly are the sequences grouped into clusters? And one way that we can mathematically quantify that is through an entropy measure that just takes the sum of p log p, or rho log rho, negative that. And that will be maximized when you have a lot of clusters of approximately the same or exactly the same size. And it will be 0 when everything is grouped into one cluster. So the coloring it in, make a tree and color it in does really well with this, because you can draw your tree like this, and you can say, okay, these look like a cluster, and these ones, and these ones, and these ones, and you can sort of tweak your method so that it will produce evenly sized clusters. The problem is you're doing that in a spurious way with SARS-CoV-2 because the tree itself isn't well supported. You can do it and you get good entropy measures. Our method achieves decent entropy measures for the 0.8 and 0.9 thresholds without having that stability problem. So yay, our method. Okay. I wanted to show you this. This is the genetic diversity in SARS-CoV-2 throughout the pandemic. So this is just a pairwise SNP distance. So that's how many polymorphisms are there between two randomly drawn viruses in the time period shown. So between January and August 2021. If you just choose two British Columbia viruses at random, you go, oh, look, they're going to be 20 polymorphisms apart, or 25. When the gamma outbreak happened, that diversity just plummeted. And that meant any clustering method that's not spurious is going to struggle to find really good or high utility clusters. You just don't have diversity there. You can't get these sequences that are all 0 or 1 polymorphisms apart and say, okay, find me the genuine underlying, really well distinguished population structure clusters that I can go and use and define outbreaks and so on, because you may have 8,000 cases. I hope you don't where you are, but anyway, we may have 8,000 cases that are so similar that they really just group into one cluster, and the utility of the sequencing is for clustering is just going to be lower. Okay, so that's a challenge. And that got us thinking about some fundamental questions in this area. Because I think what public health now wants to understand is, when is sequencing going to have high utility? And by high utility, they partly mean the ability to identify these outbreaks, distinguish them from community transmission, and proceed with these clusters. This clustering is going to work. Okay, so when does it work? I think it's a combination of 3 things. So coalescent processes that relate the virus population size to the clustering. If you have a huge population and you draw 2 taxa from it, you are likely to have 2 taxa that are quite different from each other. If you have a very small population and you draw 2 taxa from it, they're likely to be quite closely related. And that's an insight from, there's a lot more to say about it, I will spare you. But coalescent theory is a mathematical theory that helps us connect virus populations and genetic diversity and clustering. Okay, then there are several concepts related to sampling. So how many of the infections are you sampling? If you have a human-to-human infection and you sequence almost everybody that you find, and you do get good case finding, and you find, say, half the infections, then you are likely to find that A is connected to B, and B is connected to C, and C is connected to D, and and pretty soon you've got 8,000 cases, and you have this long chaining problem, which is well known in clustering, that if there isn't really a genuine underlying cluster structure, you might just find huge clusters because A, B, C, D, and so on. So that's one sampling question. And you could artificially say, well, just don't sequence so much. That would be great. But of course, there's a loss of data then, right? So that doesn't recover genuine structure. That just recovers sort of artificial structure that you would see if you didn't sample. Okay, and then there's the question of, as you are, of course, very well aware, in Australia, almost all of the transmission in the world is not in your jurisdiction. And when that changes, so that, in BC, if we had sporadic introductions from other places and little outbreaks and clusters that died out, this genomic tool is going to work brilliantly. But when we have sustained local transmission and a huge outbreak here, like we did with the P1, then when we do this clustering, we'll find this long chaining. So how many are we sampling, and what are the kind of local transmission dynamics compared to the dynamics outside the jurisdiction? And that gave us this insight that these tools of genomic epidemiology have largely emerged in a context where high-income countries have whole genome sequencing capabilities for their salmonellas and their carbapenemase producing organisms and their E. coli's and their bacterial things, and to some extent with viruses too, although that's cheaper. And a lot of the transmission of the bacteria, anyway, for which these tools are developed, is happening in low and middle income countries. And so in a jurisdiction like British Columbia, you sequence all your salmonellas and you can group them into these clusters, and this all works great. And that's partly because most of the transmission is not in the place where you're doing your sampling. So that's, I think, this kind of has been an impact of global inequality on how these methods evolved, and then what are the expectations for SARS-CoV-2. Okay, so the virus, what it's doing, the sampling, what we're doing. And then the genetic diversity. If this thing isn't evolving fast enough, then this tool will have less utility. And that's the simplest one. Okay, so that was kind of what we do with clusters. We like to estimate serial intervals because we think they're important and we think it's a new use of sequences. We use clusters to do that, but then we ask, where did clusters really come from? And I think it's moving towards this discussion. And then the next part of the talk, I think I won't really get to because we are near time. So I will leave you with one message from the What's Happening with Evolution, which is basically that in any model that describes susceptible and so on for SARS-CoV-2, we would predict that there is no direct selection happening on severity and mortality, that selection is acting on duration of infection and on transmissibility, either intrinsic transmissibility or ability to evade immunity. And because of that, that evolution is still happening, just like your coffee will still stick to the table. Next year, evolution, like gravity, will continue to be a fundamental fact of life. And I think, we need to anticipate that evolution will keep acting on this virus and unfortunately bring us potentially new variants. Hopefully this will be more mild, but there's no guarantee from the biological or modeling point of view that selection will act in that direction. Okay, so I'm going to end with thanking my colleagues who were involved in this work. I want to especially thank the Melbourne folks and Australian folks who shared this data with us, and the BC-CDC folks who shared the BC sequences with us. A lot of the math work was done by Paul Tupper and Kornia Susvitasari in my group, and Jessica Stockdale and Ben Sokoviak, also postdocs in the group who did some of this work. And opinions about COVID and its spread and speculation about what, some of the things that are more speculative are my own and are not these institutions. Okay, thank you.
Thanks. Any questions?
Hi, thanks for the talk. I'll ask a question. There's a lot here, but my question has to do with this type of analysis in the case of multiple parallel diseases that don't interact per se, flu and COVID, or other cases. Specifically here, we're also interested in these types of virtual epidemics that we release, which are kind of, digital, but they have the similar contact processes. So there's a correlation. But if you think biologically, just take the flu and COVID and say, if you're constructing these things together, can you do joint inference, tweak hyperparameters jointly, learn from one another, even though it's not on the same case, right? So you think that the person that had the flu didn't have the COVID, but dynamics are the similar. Yeah, just wondering about that.
Yeah, so I think that the cool thing to try with that would be when it gets to the virus population dynamics, because you do expect some correlation. These are both respiratory viruses, and we know, we kind of killed off flu and we really didn't have a flu season. It's going to be really interesting watching your flu season, because this is sort of the first, I guess you're starting a flu season, and we, people are writing about the immunity debt and so on for what's going to happen with flu. So I think where these tools could be used jointly would be in estimating a kind of respiratory risk in the population that would impact both COVID and flu. And then these data streams could be sort of merged to estimate that. And that might be a combination. I guess for flu, there'd have to be a term that had immunity in it, but there would also be a kind of seasonal respiratory virus or contact-driven respiratory virus term. So yeah, I think that would be exciting. You'd need good genomic surveillance. And it's not clear to me that doing this with sequences would be better than doing it with case counts. If you had good surveillance and how how many infections you have, or and you think your sampling is pretty constant with time, you might not need sequences to do it. But you could. People do argue that, oh yeah, you can get these past population dynamics from sequences. So that might be a way to join them together. With the serial intervals, or the clusters, you know, if they were both spreading in the same cluster, maybe you could estimate contact patterns. Like if you wanted to say, I had a flu outbreak and a COVID outbreak in the same complicated system, like a workplace that had a really complicated pattern. But they weren't the same people, but you had some, you know, you had, I don't know, a mining operation and you had 76 different tunnels in 3 locations and the people were moving around and so on. Like maybe you could estimate, you know, rates of risk or contact between the sub-locations or something using a joint analysis.
Yeah, great. Thanks a lot.
Any more questions?
And I'll be shorter if people want to ask more questions. I won't like blah, blah, blah for 4 minutes like I just did. Actually, it bugs me when people do that and someone asks a question and the person goes blah, 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 blah. So,
There wasn't so much blood. That was pretty good. All good.
Yeah.
Okay, good.
Thank you.
People do it though, and once you see it, you can't unsee it. So I apologize. Now you're going to see it every time anyone gives a talk.
Okay, someone commented. Robert said, thanks, Caroline, for your talk. Very interesting way to try to leverage the incredible amount of genomic data now available during the outbreak.
Thanks. Yeah, exactly. I think that's one of my kind of overall aims for the research group is, you know, we have this really high resolution data and we're analyzing it in this primarily very retrospective and focused way, but I think it can be integrated with mathematical models to try to get at prediction and forecasting or try to get at some of these mechanistic dynamics. So it's one of the longer aims.
Right. Any further questions from the audience? I think that's it from the audience. Yeah, it was great. And then we got a lot of information about the genomic analysis. And on behalf of everyone, I'd like to thank you for your time and accepting our invitation so kindly. Okay, and good to have you in our seminar series. Thank you.
Okay, thanks. I just answered one more question on the Slack, on the chat. Yeah, thanks for having me.
Yeah, thank you.